Okay, good morning, everyone. Let's begin. Just as an introduction for today, there is a lot of new content just for you. Today is the first time we've ever presented the structural variation modules. We're going to do single cell. Actually, this is the second time for single cell, so we have a little bit of practice, but there's a lot of new hands-on exercises that we've developed and designed just for you. So be patient with us, bear with us a little bit, but we're excited to teach you of some very new things today. You'll learn about mitochondria, structural variation, and single cell in the main part today. Okay, we're very, very excited to do this with you. So without further, and I will remind you at the end, fill out the survey, please, please, please. There's 2 surveys, but very short. All right, without further ado, I hand the microphone over to Aaron.
你们好. Been here 4 days. I just want to stay now. Well, as mentioned, this is actually my last presentation giving you guys today. We haven't given this mitochondrial demo a few times, but hopefully I hope to teach you a few things, like where to find some of our featured workspaces on Terra and why we even have this mitochondrial demo. So I guess you don't have to follow along with me for now, but if you do, we can just go to app.terra.bio here, right? Or like I can stay on the page. And you need to log in. I've already logged in for mine, so I guess I'm here. But if you log in here, where we want to go to today is View Workspaces. Now, you can get there in many ways. It's a nice convenient button right here that says View Workspaces. Also, you can go to what we call the hamburger menu up here and click Workspaces. What I want you to note today is the Featured tab over here, okay? We've got a lot of good stuff in this feature tab. These are where Terra keeps some of its curated workspaces that we feel show very interesting and useful things that we want people to see. And like, this is where we'll find our mitochondrial workspace. But you note, for instance, we've got the Dragon GATK one. I gave the presentation. I mentioned you can find workflows that do this. Well, I mean, here you go. It's right here. Our featured ones tend to have a lot of good description and notes, says what it does, has some descriptive pictures. It talks about instructions, what kind of inputs they require, how to run it, estimates of how much time and how much money it will cost. This is pretty standard for our featured workspaces. So I do recommend, if you all get the chance to play around with Terra a little more, to check out some of them, okay? These tend to be pretty high quality. But we're not here for Dragon GTK today. I did that a few days ago. Let's actually find our mitochondrial workspace. And Terra's thinking about it at the moment. Come on, reload. So if you go over to featured workspaces, we can search by keyword here. We're going to search for our mitochondrial one. Turns out it doesn't take many letters. And here is our mitochondrial workspace here. Note that just like the other featured workspace, it has a pretty good description of what it does. It mentioned all the workflows, which are your pipelines that are in this workspace, what data it requires as input. You've got an estimate of time and cost and all that. I mean, this is, like I said, our featured workspaces are where we put up what we consider to be good best practices examples of how you use portions of our pipeline. And not just us in GATK. Other groups at Terra will do this as well. All right. Now, it's worth asking, why did we go through the effort? Like, why do we have this mitochondrial pipeline? And the thing is, normally, mitochondrial data is actually filtered out of the normal processing in haplotype caller and MUTEC2. And the reason is because mitochondrial data is actually different in many ways. And those ways it's different actually make it hard to process in our other standard tool chains, okay? And we'll go into a lot more details about that, but we actually had to make a specialized pipeline and tweak our tools and some specialized tools just to handle mitochondrial variant calling. So like I said, we'll go into a little more detail later. But we have our data here. It is, surprise, NA12878. And if you haven't played around with Terra workflows very much, the data table shows all the metadata we have associated with there. It's kind of a wide table here, but that's okay. If you want to see what's there, you can actually just click on settings here, okay? Settings will show you all the columns of data. You can turn things off and say, oh, I don't want to see coverage metrics right now. I don't want to see these. And you can manage all the data and see what, if you want to know really what's there. It's one way to get a better view of that there, okay? The data is quite wide otherwise. And you can see we have an output VCS because I run this before. And so the output files are also here in the data table. Putting them there is nice, so you don't have to look deep down in the Terra bucket and find where they are. We often like to give links to them in the data. And also, yeah, the subset bound, which are kind of our true input to this one. So let's click over to workflows. This is our actual pipeline right here. This is our workflow, mitochondrial pipeline, okay? Now, like most Terra pipelines, there are a lot of inputs here. These are all the ways of just configuring it. Note that they all have default values. Some are pointing to data we have in our workspace. Most of them are things that we're just passing in for the alignment and variant calling. I'll talk about a little more later. A lot of MT underscore ones. A lot of them are just honestly reference files for the mitochondria, stuff like that. Note though that a lot of things are grayed out here. I can't just run it. That's because this is the shared workspace. I'm only a reader. Can I move this a little bit? That's better. I'm only a reader on this. See, read only. If you want to actually run it, you want to click the menu over here and click clone. Okay? If you clone it, you can name it. You have your own copy. And then you can run it all you want to. See, it turns out I've actually done that already. So among my workspaces, you'll see a copy of that with copy A Hatcher under it. That's me. So you see, because I'm in my own copy right now, the buttons aren't actually grayed out, right? But, I mean, instead of talking about it, let's just run it. I'll select data. We are, in fact, going to run this on our dearest friend, NA12878. Choose her sample as input. And now just click Run Analysis. Let's see. You don't have to put a comment in here. I can say, I don't know, for the, so I can type with one hand. Awesome group in Taiwan. Okay, I swear I can spell. And then launch. That's all it takes, by the way, to run a workflow in Terra. That's literally it. And this is now kicking off, and it's going to start running the analysis. Okay, you can see the status of it down here, launching. Eventually, it'll switch over to queuing and running, and you can keep track of it here. Now, I ran it again. I think I ran it again yesterday, so it might take advantage of call caching, which is nice. Call caching is a good Terra feature where if there's a particular step where the inputs are exactly the same, it'll remember that you've run that step before, and it can just retrieve your results, make it a little faster. So let's see if we actually get, I wonder if they'll get done in time. Either way, it doesn't really matter. Right now, we kicked it off, and we can move on. So I guess the question is here, I told you I'd tell you why we have this workspace in the first place. And let me hop over here and talk about that a little more now. It's out of the way. Yeah, there we go. So why? What is different about mitochondrial DNA, and why do we have a whole separate pipeline for it in a featured workspace? Well, there are a couple things. First of all, it's circular. Like bacteria and archaea, I guess, too. Well, that's fine, but that causes issues in our aligner, actually. I'll go into more detail on exactly what those issues are, but it being circular isn't a trivial difference when we're used to calling human DNA, especially because we find it with human DNA. And that's the other thing. As mentioned here, there are tons of mitochondria per cell, thousands of copies of the mitochondria. And what that means is that, so you actually have, whenever you sequence a cell, by the way, and whenever you have any kind of human data that comes out of there, you're also going to get mitochondrial data, like it or not, right? And the harder thing about mitochondrial data, too, is that how much is there relative to your standard autosomal DNA actually depends on the tissue type. In the mature red blood cells, I mean, whatever, you don't have many there, but in muscle cells, you got a lot. In liver cells, you have tons of mitochondria. So because of that, when you sequence like a standard piece of a standard tissue sample and you want to actually get your nuclear DNA, you might have 30x coverage there, fine, but you might have thousands, tens of thousands of copies of the mitochondrial DNA. And that matters. We have huge read depths. And if you want to do a deep coverage of your autosomal DNA, you might have massive coverage of the mitochondrial DNA. This is a problem for our tools. I mean, if there's any team that would know that it's my team doing GVS, sometimes just throwing more data at something can make it a new problem. And of course, another issue with that is haploid. You don't have the standard 2 copies of it. You got one, you got them from your mother. This can be a bit of a problem in the haplotype caller. It makes some assumptions about being diploid there. So the ploidy assumptions get a little difficult for our tools to handle naively, at least. Luckily, what we often want to look at, we're not as interested most of the time at looking at what copy of mitochondria did we get from our mother. We often want to know some of the somatic changes, things that we've acquired in our lifetime to our mitochondrial DNA. And because of that, okay, we look at maybe use Mutech. And that's a better fit than the bare haplotype caller. The thing is, Mutech, as we mentioned, because it's used to sequencing tumors, it doesn't make strict assumptions of ploidy or anything, right? So it's a good tool for figuring out like low frequency alleles. But the problem is mitochondria are weird in yet another way. You've got these wonderful things, a new source of contamination purely when doing mitochondrial DNA called new mites. So basically, over evolutionary timescales, little bits of mitochondrial DNA have been absorbed into your nuclear DNA. They're in your autosome now, right? And it's a problem because there are a lot of them all over, and the amount varies. Some are maybe 1,000 bases. You've got some places where you've got basically upward of 16,000 bases. The entirety of it is just sucked in there. And what happens? Like at some point in history, some cell had a really bad day. And something happened, it panicked, it's putting itself back together, it slurped in the mitochondrial DNA, and that's just part of you now. And to make it worse, the amount actually varies by population. Like other things, the exact amount you have of these new mice varies. So again, it can be a little hard to deal with. And because they're no longer in the mitochondria, being functional, producing useful proteins, they're actually decoupled from a lot of selection pressure. That means they get to acquire neat and wonderful mutations. There's no constraints, right? If they don't work, who cares? They're not even functional now. And maybe we wouldn't care, except they often get aligned to the mitochondria because they look like they're from the mitochondria. They are from the mitochondria. They're just not currently residing there, not currently function. Now, as you can imagine, this produces a ton of artifacts, right? Now, not only do you get like alignment issues, you get downright errors sometimes, and you'll see things, just noise. You'll see mutations there that are present in these new mites in your nuclear DNA that are irrelevant to the functioning of your cell. Like they're not actually being transcribed and doing anything in the mitochondria. They're just noise. So when you're looking at things like, you want to look at subclonal fractions and look at maybe low frequency alleles in there, this is a problem because you'll have these in there and they're going to add their own unique type of noise to your data. So what does our pipeline look like though? Let me move this window again. So we do have a whole pipeline though. That's why we have to have a separate pipeline for analysts. Well, the first steps are pretty easy. You've got subset BAM, revert BAM. All right, I mean, that's kind of a subsetting everything. We only want to look at the reads that are on the mitochondria. This is the mitochondrial pipeline after all, right? But reads that are on the mitochondria, or as I mentioned with new mites, look like they're actually in your mitochondria. We'll subset to those. We actually revert the BAM after that and do a full realignment. And now we have new steps you can see here. You've got aligned MT and aligned to shifted MT. That is where we actually try to align to our reference. Now, remember I mentioned mitochondrial DNA being circular causes a problem? when you're trying to lay out, the bases on the chromosome, you pick a start, you pick an end, and you align to that. Well, there's no real start or end. I guess that's just a circle thing, kind of by definition. So the problem is, how do we handle that? To some degree, okay, you pick an arbitrary point, right? In our reference, we cut there and say that is 0, and we just number our way around. Simple enough. The problem is that has nothing to do with nature. And even when you're reading it, your reads don't care that we start numbering at a certain point. Wherever we make that cut, we start having big alignment issues there. We have reads that will cross that boundary because as you're reading around, it just keeps going, right? So on those edges, we have massive errors. I mean, they can downright blow up some of the aligners. So what we actually do here with align to shifted MT is it's a simple solution. We produce 2 versions of the mitochondrial reference. Cut it in the first place, and we actually do another version where we cut it the whole way around, on the other side of the circle, 180 degrees, and we cut there. Why? We align to each separately, and yes, we'll have issues on the edges, but if you combine the information from these 2 separate BAMs, turns out you can actually get a good read for the entirety of the mitochondrial DNA. And that's why you see the aligned to shifted MT part there. Now, we said we use a special version of MuTech2. Remember, MuTech2 seems like a good tool for this because it doesn't make any kind of ploidy assumptions, and it can handle some low frequency alleles and everything. We have to modify it some. It takes a couple new arguments for handling just the sheer depth of data we're dealing with. It's got to be modified a little bit. But also, so as I mentioned, the depth can get crazy on mitochondrial DNA. Like, it's one thing to look at a 30x coverage in a genome. It's another to have, I don't know, 25,000 depth. And things happen when you have that many reads. Weird things happen. Like, when you think about 1 error per 1,000 bases, just in sequencing, how does that add up when you have 25,000 separate reads? The sequencing errors are no longer a trivial fraction. They become significant. And that makes it very difficult when we're trying to go in and align things and reconstruct the graphs. You get crazy graphs if you aren't careful, because there's a lot of noise in there all of a sudden. Just that amount of reads means that just errors pile up. You start getting these noisy graphs. So there is some tweaking we also had to do to MuTech2 to be able to handle that. And we also mentioned the new mites. Well, there are special filters in there. One of the tricks to try to filter out most of the new mites is we did have from the original files what the coverage depth was from the autosomal DNA. We have that. So any variants we see that occur with roughly that allele frequency, that aligns the coverage depth of your nuclear DNA, we just kind of have to assume that they probably came from the nuclear DNA. That's one of the ways we get around the new mite problem. Of course, it means we'll miss things. There might be legitimate alleles that happen to align, and we happen to get them with that coverage, but we can no longer be sure. But eventually, okay, we're done. We call our variants using our modified MuTech. We merge, do some filtering, and really that's it. We have our variant calls. Yeah, mitochondrial, it has its own complications, but in the end, the pipeline is relatively fast, cheap, easy to run, and fixes the issues that you might have using an unmodified sort of naive haplotype caller to attempt to do this kind of calling. All right? Let's head back to Tara for a second and see if our call is done. It's done. Running. Well, that's okay. It turns out, like I said, I have a version I ran yesterday anyhow, just so it's done in time. What they do in cooking shows, right? They put it in the oven and they magically reach out, and here it is. So this is my freshly baked bread right here. So you can dig in. It says it's done. You can look at the inputs for it, how it was run, and the outputs. You can click over. You can get a job manager. Probably prompt me to log in. Let's see. Also, I'll point out that this pipeline is deterministic. Okay, so it doesn't. You run it twice, you'll get the exact same answer. So we can look at the one that I ran yesterday and have some confidence in it. And when you look at the data, you can see a lot of what's going on, all the individual tasks that were in there, inputs passed, you can actually check per task. Outputs, for instance, here's everything the pipeline sped out. You've got like your output VCF. Remember I mentioned the really, really kind of long and weirdly named Google buckets that Terra makes for you. You can see that, the FC blah, 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 blah. Yeah, that's the spot in your Terra bucket where it dropped it. Don't worry, you'll never have to know that. But you can see it here in the outputs, okay? If you care to analyze it more, you can look at the timing diagram. You can see where it spent its time. What parts were actually did it spend its time churning on? There's a bunch of information here, okay? But it's done. It's done running. So let's this out of the way. Let's get back and view our data a bit. All right. What came out of this? We're going to use the web version of IGV here. Okay. We use the web version in several other demos. It's convenient when you just want to take a quick look at your data. So let's load it up here. 812878. Open with, out the way, IGV. We'll look at the BAM, the realigned BAM, and the final VCF. Yep, HG38. And we'll launch it. Now, the same things that make it hard to sometimes call variants on mitochondrial DNA can also make it hard to view it here in IGV, which is to say you have a large read depth sometime, okay? Now we're going to try to zoom into a chromosome M250. That has a known variant. Yep. Good thinking. So how's the weather? I'm not going to make you wait for this because, ta-da, I opened it in advance. It doesn't take that long, okay? But I'm not going to waste your time. So what can you view in here? You can view the call, right? You can view things like, all right, you can see it called a variant there, just like normal IGV. You can click on it. Okay, the ref was A, the alt was G. And you can see data here about like strand bias and what it calculated for that. Pretty balanced. See why we called it. So you can also, let me see. Yeah, you can also, when you're looking at this, you see the whole genotype. Is actually A and G. There's some phasing information there. It's the pipe means. Look at the depth there. You got a 10,000 read depth. Yeah, it's mitochondria, right? But the funny thing is, if you go up here to the original BAM and you look at that site, you actually had 25,000 here. We actually downsampled a bit when we were making our call. Like I said, you get a lot of stuff. Now, the good thing is, all right, you can actually get pretty solid information on some of these real signals, right? From like subclonal populations, because we have so much data in there, you can get good evidence for them. It's just handling all that data, that's kind of the issue, okay? But my point is, you can use IGV to view the stuff just like you would anything else, all right? So, I guess in summary, what's good about data, about mitochondrial calling? Like I said, you actually have some good confidence in things when it comes out of this. That massive depth, like I said, it's good. When you see something there, you're pretty good. You figure out the pneumites, okay, they complicate things, and you got to kind of do the alignment, but the pipeline does that for you. But because of that large depth, you have to be careful in how you process it, okay? Again, with that many reads, you're going to have issues with just the natural, relatively low 1 in 1,000 baseline error rate of the sequencing. It piles up. So a naive caller might not be able to handle it. They can just downright explode. But that's why we gave you the special version of Mutech in here. And you're welcome to clone the workspace yourself, play around with it a little bit, and see what you might find. That's actually all I have today for the demo with you. Any questions? What's up?
Very small particle, the flow exome. So the regular metric dissect is not fit for 500 February.
So the question was because the mitochondria is such a small fraction, of the total DNA, the regular QC metrics that we use on calls might not be fit for them. And should we take metrics separately on the mitochondrial DNA? It sounds like a good idea, but I actually don't know what they do in practice for this. Do you know what they usually do, Eric? Mm-hmm. So the answer Eric gave was that partly because this is not diploid, remember, a lot of the QC metrics we talked about before don't really work for this, right? So we actually have a whole separate set of QC metrics that we have just for mitochondrial data. Another complication is that there's not as much truth data for it, but we do have a whole other set of metrics that we run for just the mitochondria. That's a very good question. Do we have any other questions? All right, awesome. In that case, I imagine we can move on to our next presentation. Who's up next? You're lucky you get Emma. Not that any of us are unlucky, really, but. Awesome. I'm paranoid turning it off and putting it down because sometimes it got. I'll be your microphone holder for a little while. Okay, I can still be functional. I hold a good microphone.
All right. Thank you.
You're welcome.
All right, well, this is the moment that I've been waiting for because I work on structural variants, and now I get to talk about structural variants. So in this session, this is the very first time that we've presented this session, so that's exciting for me. And I will be talking about our pipeline for characterizing structural variants from short-read whole genome sequencing data. And that pipeline is called GATK-SV. So I'll start by giving a little bit of background. Some of this will be review because we've already talked about copy number variants. So let's start off with defining structural variants. So a structural variant, abbreviated SV, is a DNA sequence alteration exceeding 50 base pairs. And this figure shows an example of a structural variant, specifically a deletion. So we can see that the sample genome on the bottom is missing the light blue region that is in the reference on top. And the junction where there's a break compared to the reference is called a breakpoint. And all SVs are composed of at least one breakpoint. And that's a term that I'll use throughout the talk. So beyond just deletions, there are many types of structural variants. Your DNA can get rearranged in a lot of different ways. So we'll start with copy number variants because we've already seen those before. So a reminder that those include deletions or duplications of a stretch of DNA. And that also includes multi-allelic CNV sites where there's a lot of different copy numbers that we observe in the population. Below that, we see a category called balanced SVs. So these are structural variants that do not involve a gain or loss of a copy of DNA. And these include insertions, where there's a novel sequence that gets inserted into your DNA. Inversions, where a section of DNA gets flipped around in its orientation. And translocations, where whole pieces of chromosomes get moved around and merged back together in ways that they were not originally put together. In the top right, then we have complex SVs, And these are structural variants that actually contain more than one type of SV signature. So for example, a biological event that's one single biological rearrangement, but we see both a duplication and an insertion signature, for example. And I'll talk about those in a little bit more detail later on in the talk. And finally, on the bottom right, we have break ends. So we sometimes, from the GATK-SV pipeline, output break ends when we find some kind of evidence that there is a structural rearrangement happening at some position in your genome. But based on the evidence that we have from short read data, we're not able to fully resolve what type of structural variant that is. But we still want to tell you that there might be some signal there. And we talked yesterday about the role that copy number variants play in disease and your characteristics and the evolution of humans over time. But beyond just copy number variants, other types of structural variants are also really important in disease. So looking at balanced and complex SVs, because we've already talked about copy number variants, there are a whole bunch of studies that have linked pathogenic, balanced, and complex SVs to phenotypes that range from congenital skeletal defects to neurological disorders. So really severe disease phenotypes have been associated with these balanced and complex structural variants. So it's important to characterize the full spectrum of SV types when you're trying to understand human disease. Okay, so now that we've defined structural variants, let's talk about how they show up in short read whole genome sequencing data. Typically, as we mentioned during the long reads presentation, short reads at about 150 base pairs long are often too small to contain an entire structural variant, which could be thousands or millions of bases long. So we can't just read off the structural variant directly from the read. We can't just look at the cigar string like we might be able to do in long read data. So instead, we use 4 different types of evidence: split read, paired end, read depth, and B allele frequency. So now let's take a look at each of those 4 types of evidence in more detail. Let's start with split read, or SR, evidence. So we have 2 samples shown here. Sample 1 on top does not contain a structural variant, so it kind of matches the reference, and each of the reads in black align back well to the reference. But sample 2 on the bottom has a deletion of a segment of DNA in the middle there compared to the reference. So when we sequence sample 2, the reads don't align back perfectly to the reference. We see that part of the read shown in black aligns back just fine to the reference. But then we have this red half of the read, and that's called a soft clip. And that's where the read does not match the reference at that position. And that's because the sample doesn't contain that deleted sequence, but the reference does. So the red side of this read is actually over here. But when we align it back to the reference, that's not where it gets put. So it just doesn't match the reference and it gets soft clipped. And that's what we call a split read. And you can see here that where the soft clip starts actually tells us the precise location of the breakpoints of this deletion. So split reads are a really powerful type of structural variant evidence because they give us such precise breakpoint information. So let's take a look at how this shows up in a real life example in IGB. So this is an example of a heterozygous deletion, and on the right, I've zoomed in on either side of the, or rather on one side of the deletion, and we can see this rainbow of colors in several of the reads, and that indicates the portion of the read that is soft clipped. So we see that visually in IGB, we can tell that there's split read evidence for this heterozygous deletion. Okay, so now let's talk about the next type of evidence, which is paired end or PE evidence. So remember that with Illumina sequencing, we sequence from both ends of the sequencing fragment. So we end up with two paired reads, two mates that came from the same sequencing fragment. And they don't overlap. They don't touch each other in the middle. But we know that they are typically oriented inwards because we sequenced from either end. And we know that the typical insert size, so the distance between the start of each of those mates has a typical value. So they're typically closely distributed around the mean insert size. We have an expectation for how far apart those mates should be. But when we see deviations in the insert size or the orientation of the reads, that can indicate the presence of a structural variant. So, for example, we see that with this deletion, the insert size is larger than we expect. And with this tandem duplication, the orientation of the mates is now outwards instead of inwards. So let's take a little bit of a closer look at what's happening there. So in sample 1 on top, there's no variant, and each pair of reads aligns well to the reference. They're inwardly oriented, and they have about the same expected insert size. But sample 2 has a deletion compared to the reference, and we see that the alignments, the insert size is farther apart than in sample 1, so farther apart than the expectation. And this is because when we created sequencing fragments, when we sequenced sample 2, the deleted segment is just not there. But when we align those read pairs back to the reference, suddenly we've added that deleted segment. And so the reads from either side of the deletion get pushed farther apart. And then we see that the insert size is larger than the expectation. So paired end evidence is very powerful in terms of giving us a signal for structural variance, because there are different patterns of orientation and changes in insert size for different types of SVs. So it's giving us a lot of information about the different SV types. But it doesn't tell us the exact location of the breakpoints. You can tell in this example that we can see approximately the location of where there should be a deletion, but you can't tell just from the paired end evidence exactly the base pair location of where the breakpoints are. Okay, so let's go back to our real life example in IGB. And we have colored and sorted by insert size. So we can see that there are discordant pairs in red towards the top. And we see that the insert size is much larger for those red pairs of reads than the ones in gray. Give you a moment to look at that. So we can see visually in IGV, with a little bit of help from coloring and sorting, the paired end evidence for this heterozygous deletion. Okay, next one is read depth or RD evidence. We've already talked about this when we talked about GCNB yesterday, but I'll review it. So basically, changes in read depth can signal the presence of copy number variants if they're large enough copy number variants to actually impact the read depth. So we have a few examples here. On top, we have a heterozygous deletion, and we see that the coverage over the deleted segment is about half of what it is on either side of the deletion. For the homozygous deletion example, there's a complete lack of coverage over the deleted region because that region just doesn't exist in that sample, so we can't sequence it. Then we have an example on the bottom of a heterozygous duplication, and we see an increase in coverage over the duplicated region because we have more copies than usual to get sequenced. And again, we can see this when we look at our real life example in IGB. So the coverage track is above the reads, and you can see that there is a drop in the read depth over the deleted region. So if you look for the red paired end reads, right above that, there's a drop in the coverage. So again, in IGB, when we inspect this variant visually, we can see the read depth evidence here for this structural variant. Okay, our fourth and final evidence class for structural variants in short read whole genome sequencing data is B allele frequency, or BAF. So the B allele frequency or allele fraction for a heterozygous SNP is typically around 0.5, because if it's heterozygous, the SNP is on one copy but not the other, so you expect about half of the reads to have that SNP. So you get an allele fraction of 0.5. But when there are copy number variants, that allele fraction can change. So in the first example, we have a heterozygous deletion. So one copy of this segment of DNA has been deleted, and on the other copy in the deleted region, we have two SNPs, and the SNPs are shown by the yellow stars. And so when we go to look at our read alignments, the allele fraction, instead of being 0.5, is actually 1, because we only have one copy of that stretch of DNA, and the SNPs are on that one copy. So the allele fraction is 1. Okay. Our second example is a heterozygous duplication. So on one copy of this region of DNA, there's a duplicated segment, and on the other copy, there are two SNPs. And when we look at the allele fraction for those SNPs, it's about 1/3, because they only occur on 1 out of 3 copies instead of the normal 1 out of 2. So we can see how changes in copy number can alter the B allele frequency for heterozygous SNPs. But this is kind of a fuzzier evidence type, like where can you draw the line between 1/3 and 1/2? It's not going to be exact every time. So in the GATK-SV pipeline, we actually only use the B allele frequency for variant filtering rather than variant discovery. Okay, so let's take a look one last time at our real life example of a heterozygous deletion in IgB. So we can put all of these pieces together. So there are actually some SNPs in the deletion region. And if we go up to the coverage track, we can see that each of those SNPs only has one color. So that tells us that the allele fraction for each of those SNPs is 1. So in that way, we have support from BAF evidence for our heterozygous deletion. And now we have observed a support from all 4 types of SV evidence for this heterozygous deletion. So we really believe that this is a true deletion based on what we've seen here. Okay, so now that I've talked about how structural variants appear in short read data, let's talk about the pipeline that we use to detect them computationally based on those 4 evidence types. So the pipeline is called GATK-SV, and it is a computational pipeline written in WDL for the discovery of structural variants, germline structural variants from short read whole genome sequencing data. And your inputs to this pipeline are going to be Illumina short read deep whole genome sequencing data, so BAM or CRAM files aligned to HG38. And your outputs are going to be a genotyped and annotated joint SV VCF, fluidity estimates, and supporting QC reports. And similar to GCNV, there are 2 modes in which you can run GATK-SV. The first is cohort mode, where you run SV calling on a group of at least 100 samples, and that produces a joint SV call set. The second mode is single sample mode, where maybe you don't have 100 samples and you want to run SV calling on just 1 sample at a time. But this mode allows you to still take advantage of the joint steps of the method by using pre-computed data from a reference panel of samples. So I'll talk a little bit more about how to run GATK-SV in single sample mode later on in the presentation, but most of the methods will focus on cohort mode. So just a little bit of history. GATK-SV was originally developed by the Tukhowsky lab at Massachusetts General Hospital for studies of autism spectrum disorder. It was later refined and optimized and scaled further to be applied to the Genome Aggregation Database, or GNOMAD, to produce a population reference of SVs in a human population. And so the figures on the right are from the GNOMAD SV paper on about 14,000 samples. And I won't go through those figures in detail, but the paper is pretty cool if you want to check it out. And the figures show the diversity of the samples in the call set, as well as the sensitivity of that call set. And the Tukhowsky lab continues to apply GATK-SV to larger and larger groups of samples to create better human SV population references to be used in research. So a couple of exciting ones that I want to point out are the latest release of GNOMAD. So GNOMAD v4 contains SV calls on about 63,000 samples. So that's a lot bigger than the previous release of 14,000. And that was released in November of this year. And then there's also SV call sets for the All of Us research program. We've already released one on about 11,000 samples. And early next year, we plan to release one on almost 100,000 samples. And as part of applying GATK-SV to samples from Nomad, the performance was heavily benchmarked. So in terms of orthogonal data sources, there was a 97% molecular validation rate of de novo SVs, 97% sensitivity to CNV calls from microarrays, and 94% confirmation rate by long-read PacBio sequencing data. So for all of these comparisons to orthogonal data types, the number that we're looking for is really 100%, and we got pretty close for all three of those. Next, in families, we observed about a 4% Mendelian violation rate. So the number we're looking for there is close to 0%, because there is a very low rate of de novo SVs per individual, less than 1. But 4% is about on par with SNP and indel callers, actually, so we're still doing pretty well here. About 86% of SVs were in Hardy-Weinberg equilibrium. So in a theoretical population, we'd want that to be close to 100%, but there are other reasons besides errors in genotyping that could cause that to be lower than 100%, such as population stratification or natural selection. So we don't expect to get to exactly 100 there. So 86 is pretty good. There was also strong linkage disequilibrium with nearby SNPs and indels. The gold standard value is about R squared equals 0.8 for this, and we got above that. So we're pretty happy with all of these results. And we think that GATK-SV does a pretty good job of producing a structural variant call set that's both sensitive and specific. Okay, so let's talk about how we do it. In this methods section, the key concepts that I'll go over are the ensemble approach of GATK-SV, variant merging, joint genotyping, complex event resolution, functional annotation, and quality control, or QC. So I'll start with a schematic of the pipeline to see how all those pieces fit together, and then I'll go through each of them in more detail. So GATK-SV takes an ensemble approach, applying 5 different SV discovery algorithms and taking advantage of all 4 of the evidence classes that we discussed. Then we perform variant merging before performing joint genotyping of all sites across all samples. We resolve complex events. We perform functional annotation. And then we use QC to verify that we've produced a high quality call set. And the GATK-SV pipeline is very computationally intensive. It's highly parallelized. So it is designed to run in Google Cloud, and we've set up Terra workflows in order to make that easier. But unfortunately, we don't currently support any other backends besides Google Cloud. Okay, so evidence classes. We went through the 4 different types of evidence. That was split reads, paired end, read depth, and B allele frequency. And we kind of saw how each of those evidence types has its own strengths and weaknesses. So GATK-SV uses all 4 evidence types to get all of those strengths together. But there isn't a single algorithm out there for SV discovery that actually takes advantage of all 4 evidence types at once. So because of that, GATK-SV runs multiple SV calling tools and combines the results. So in that way, GATK-SV takes an ensemble approach to SV calling for optimal sensitivity and specificity. So these are the tools that we run for SV discovery. First is MANTA, which is a really good all-purpose SV discovery algorithm. Then there's MELT, which specializes in calling mobile element insertions. And then there's WHAM, which we've found has higher sensitivity to small duplications that are missed by other algorithms. So we run multiple tools because they each do better at different things. And we want to do as well as we can on everything. But none of these three tools use read depth evidence. So on the next page, I'll show you the tools that we run that do use read depth evidence. I'll also note that MELT requires a license to run, which we don't really like because we want all of our code to be open source. So we're working right now on switching over to a new mobile element insertion caller called Scramble, which is open source. Okay, so these are our CNB callers that use read depth evidence. So the first one should be familiar. It's GATK GCNB. We mostly talked about running GCNB on exomes yesterday, but we do run it on genomes as part of our GATK-SV pipeline. And the second one here is CNMOPs. So both of these detect copy number variants using changes in read depth, and both of them are joint callers, so they require a group of samples to run. And using both of these methods together improves our sensitivity to CNBs and our segmentation, so the determination of CNB intervals. Okay, so after we have performed SV discovery using all 5 of those algorithms, there's actually going to be a lot of redundancy. We've run 5 different SV calling tools on each sample, and we have many samples as well. So it's very likely that the same SV has been discovered by multiple algorithms and in multiple samples. Also, structural variants are not always determined with precise breakpoint precision in short read data. So we expect there to be some variation and uncertainty in the exact start and end of the SV interval. So for those reasons, if we see a bunch of similar overlapping variants, like the variants in black that are almost over the same region but have slightly different start and end, we actually assume that those are representing the same underlying structural variant. So what we do is we cluster those together and we collapse them into a single representative variant to reduce redundancy in our call set. After that, we can proceed to joint genotyping, where we assign a genotype at every site in every sample based on the PE, SR, and RD evidence. So in our table, we can see that every sample has a genotype at every site, and that can be homozygous reference or 0/0. It can be heterozygous or 0/1, or it could be homozygous alternate or 1/1. And similar to with SNPs and indels, we get a lot of benefit from performing joint genotyping, both in sensitivity, because we might be able to rescue SV calls that were missed in one sample, but discovered in another sample by the original SV discovery algorithms. And we also see gains in specificity, because for each site, we can learn the distributions of the RD, SR, and PE evidence at that site, so we can better distinguish between carriers and non-carriers at that site. After joint genotyping, we want to resolve complex variants. So these are there are many different types of complex variants. Remember, that refers to any structural variant that actually contains multiple different SV signatures in one unified biological event. So you can imagine that these things get complicated, and there are a lot of different patterns of complex SVs that we observe in human data. But GATK-SV resolves a subset of those types of complex SVs because we have to know what to look for. So we resolve these 11 subtypes of complex events. I know it's a little bit small, but you can also look at the Nomad SV paper to see this in more detail. And I'll just talk about one as an example. So on the bottom here, we have a dispersed duplication. So this is where a segment of DNA gets duplicated, and that extra copy gets inserted somewhere else in the genome. So it's a copy and paste insertion. And that means that there's actually a duplication signature as well as an insertion signature for that event. So that makes it a complex event. And if we just look at each component SV signature of a complex event, we're not going to get the full picture. We're probably not going to be able to interpret it accurately, for example, in a clinical context. So resolving these complex events and understanding how the pieces fit together is really important for downstream variant interpretation, as well as furthering our understanding of how structural variants occur biologically. Next, we perform functional annotation using the GATK tool called SV Annotate. And the questions we're really trying to understand here, the questions we're trying to answer here are what genes or non-coding elements are overlapped by our structural variant, and what impact do we expect the variant will have on the protein function or transcriptional regulation? So what is this SV going to do biologically? So I have a couple of examples for you to illustrate this. Our reference sequence is on top, and our alternate sequence, or our sequence from our sample, is on the bottom. And we can see that there's been a deletion of a single exon in the sample relative to the reference. And SV Annotate will predict the consequence of this event as loss of function, because we have removed a whole chunk of the protein, and that's probably going to impact the protein's ability to function. Another example here is a whole gene inversion. So the breakpoints of this inversion are outside of the gene boundaries, and the entire gene's orientation has been flipped. And the annotation from SV Annotate in this case is that it's a spanning inversion. So this one's a little bit more descriptive than predictive of a consequence. And that's because it's actually a little bit unclear what biologically the consequence of this event will be. The entire gene is still present and intact. It's unbroken. But flipping it around in this way could have disrupted its transcription if we disrupted the regulatory elements on either side. So we want to let you know that it's a spanning inversion, but also that there might be some uncertainty in how to interpret that. So those were just two examples to illustrate what we're trying to do here. But you can imagine that there's a lot of complexity to this problem in terms of how each different type of structural variant overlaps with the different elements of a gene, the introns, exons, the UTRs. So as a result, there are 17 different classifications that SV Annotate produces. 13 are predicted protein coding consequences, so something that is happening to a protein coding sequence. And 4 are non-coding annotations, so something that's happening to a non-coding element. And when we plot how many of each annotation we get on a sample BCF, the most common annotations are going to be intergenic and intronic. which makes sense because a majority of the genome is non-coding. Okay, so our last step here is performing quality control on our call set. And GATK-SV produces a bunch of metrics and plots to help you understand what your call set looks like. So this is an example of one plot that's produced, which shows the number of SV sites per genome. On the left, it's just all SV sites, and on the right, it's broken down by SV type. And what we're looking for here is that we expect about 7,000 to 10,000 SV sites per genome if we exclude these BNDs over here. So remember, those are unresolved break-ins. We don't know exactly what's going on there. So typically we remove them for some of our analyses. And if we exclude those, we expect to find about 7,000 to 10,000 SVs from short read data. And that indicates reasonable sensitivity. In terms of specificity, in cohort mode, we do recommend performing additional downstream filtering if you are doing this for an application that requires high precision. And we have developed machine learning models to perform filtering, and we're working on releasing those publicly. Okay, so in this methods section, just to recap, the key concepts that we talked about are GATK-SV's ensemble approach, variant merging, genotyping, complex event resolution, functional annotation, and QC. So once you have run GATK-SV, how can you understand and interpret the SV calls that you get as output? So GATK-SV produces SV calls in VCF format. And here I'm showing an example of a structural variant that was from a study in the Kovalsky lab of the genetic causes of disease for fetal structural abnormalities. So they were looking at causes for very rare diseases in this study. And this is just one record from a VCF, so one line of the VCF, but I've broken it up across multiple lines to make it a little bit easier to read. And I've also added the header for each column so that you can tell what you're looking at. Okay, so let's walk through what you can tell from this VCF record. First, where is this structural variant? It's at this position on chromosome 12. We can see that in the chrome and pause fields. But remember that a structural variant is an interval with a start and an end. So the pause encodes the start position of the interval. And then in the info field, we have the end. End equals a bunch of numbers that I'm not going to read. And then what is this structural variant? So we can find the SV type in two places. There's the alt allele, which is a symbolic allele that says del. And there's also a field in info that's called SV type equals del. So this is a deletion. At the same time, we can look in the info field to find the length of this SV. So SV len equals 1369. So this is a 1,369 base pair deletion. Then we want to know what's going on in our sample of interest, which I've called sample X. So we can look for the genotype of this sample. So we can see way down at the bottom that the genotype is 0/1. So our sample X is heterozygous for this deletion. We can also see which evidence types were used to determine that genotype. So what evidence was supporting the presence of this deletion in this sample. And we can see that it actually had support from RD, PE, and SR evidence. Okay, so what is the quality of this structural variant? We can look first on a site level. What is the quality of the site in all of the samples? And for that, we would look at the filter field and its pass. So a filter has not been applied to this site. And we can look at the qual field, which is 50 out of a maximum of 99. So it has, medium quality. Then we can look at the quality in the particular sample. So way down at the bottom again, we can see that the GQ is 49 out of a maximum of 99. So quality is good, it's decent. We can also see genotype qualities for each of the 3 evidence types that supported this genotype in this sample. And so it's a little hard to tell with just all of the colons and everything, but actually we can see that the genotype quality from PE evidence is 89. So there was very high paired end support for this deletion in this sample. So the quality here is pretty good, but actually let's take a look at this in IGB to reassure ourselves. So I've highlighted the deletion interval in red here, and we can see that there is, if we look at the coverage track, there is a bit of a dip in coverage. It's subtle because this is a heterozygous deletion, but it's there. We can see that there is read depth evidence. If we look on either side of the deletion interval, if you look really closely, you can see that there are those rainbows of mismatches that indicate a soft clipped portion of a read. So there is also split read evidence for this deletion. And finally, the discordant pairs are colored in red. They're not sorted as nicely as they were in that first example IGB screenshot that we looked at, but they are there. So that shows us that there is paired end evidence for this deletion. So after looking at IGB, I trust this variant call. It looks good. We can see all of the evidence that supports this genotype. Okay, so maybe our next question, since we're interested in genetic causes of rare diseases, how common is this variant? So we can go to the info field, we can look for the allele number, or AN, allele count, or AC, and allele frequency, or AF. And we can see that the allele count is 1. So actually, this variant was only observed 1 time in a heterozygous state in this entire cohort, which had about 900 samples. So the allele frequency in this cohort is quite rare. It's not shown here, but we could also annotate this structural variant with the allele frequency in NOMAD, and that would tell us from a larger set of samples how rare is this structural variant. So just not in this example. Okay, and then we want to know, is this variant clinically interesting in terms of its functional consequence? So SV annotate has told us that there's a predicted loss of function in the gene MED13L, which turns out to be associated with developmental disorders that are consistent with sample X's phenotype. So taking all of this information together, that this is a pretty high quality structural variant. It's rare, we don't see it in other individuals, and it is predicted to cause loss of function of a gene that has already been associated with a disease that's similar to the patient's phenotype. All this together, this is a pretty strong candidate to be the cause of disease for this sample. Okay, so hopefully I've kind of convinced you that GATK-SV is a useful pipeline. Maybe you want to run it. So how can you do that? So we're going to talk about running GATK-SV in single sample mode. So this is where you run GATK-SV on one sample at a time. And it's recommended for small sets of samples where you don't have enough samples to run GATK-SV in cohort mode. So you have fewer than 100 samples. So this is useful for rolling sample sets. So in a lot of clinical settings, you don't get all of your samples at once together at the same time. You just get a few at a time. And so it's much more convenient to be able to process one sample at a time. The single sample mode is faster and easier to run than cohort mode, but some caveats: it's more expensive per sample and it has a little bit lower sensitivity. And the single sample mode requires pre-computed reference panel resources that were generated by the cohort mode of the pipeline. And we actually provide pre-computed reference panel data from samples from the 1000 Genomes project that are publicly available. So you can use that reference panel data if your data is similar in terms of, the sequencing depth and everything to the 1000 Genomes panel data. And finally, the single sample mode of GATK-SV is available publicly in Terra as a featured workspace. So you can go to Terra right now and find this. The cohort mode, we're working on getting our workspace featured, but it's not quite there yet. So we're hoping to make that public very soon. Okay, so how do you run GATK-SV in single sample mode on your own data? First, you go find the featured workspace. You can see the name of the featured workspace in this screenshot if you want to go back to it later. And then you make a clone so that you can actually make changes in that workspace. Then you upload your sample metadata. You really just need a sample ID and a BAM or CRAM file. You also need this Boolean, is the BAM or CRAM file in a requester pays bucket. That's just a specific type of bucket in the Google Cloud. And then there's just one workflow to run, and all of the inputs and outputs are pre-configured to refer to the data table. So all you have to do is launch this workflow on your samples. It will run for somewhere around 24 hours. There's a lot happening in this workflow, and we expect the cost to be around $7 to $10 per sample. Your outputs will include SV calls in VCF format, as well as in BED format, and metrics and QC plots, as well as ploidy plots that show larger scale copy number information. Okay, so that's basically all I have, but I wanted to provide some resources for further reading. So we have resources on the background of structural variants, as well as the Nomad SV paper. We have resources on the GATK-SV methods, and resources on how to run GATK-SV on your own data, including troubleshooting for common errors. And that's all on GATK-SV. Any questions? Yes.
If you run the SV calling on a single sample mode, can you still get the allele frequency record?
Yeah, so good question. You can get the allele frequency with your sample and the reference panel, and you can also annotate the allele frequency from another call set like Nomad. So those are the two ways that you can get allele frequency in single sample mode. Okay.
Is there a detection limit in structural variant discovery? For example, can we discover very long deletion, for example, a megabase or more than that?
Yes. So the read depth evidence is very good at discovering very large copy number variants, including ones that are a megabase or longer. Yeah.
There is a type of structural variation called large genomic rearrangement on the BRCA. Do the GATK-SV code BRCA mutation?
I believe it should. I haven't looked specifically for variants from GATK-SV in BRCA, but if it's an SV, we should be able to find it, unless it's in a particularly repetitive region that would require long reads. Oh, okay.
I have a question of that VCF example. So you mentioned that there are 5 algorithms used in GATK-SV pipeline, but why does it just show Manta as algorithm?
Yeah, so in the VCF, you can actually see which algorithm originally discovered that variant in that sample. And so for the example that we were showing, Manta was the one that found that variant.
Is there is it GATK-SV possible to detect the translocation, or is there any recommend highlight to observe that?
Yes, GATK-SV can detect translocations, although I would recommend really scrutinizing those translocation calls because they're pretty rare in the population. So you want to make sure that we got it right.
I have a question about the merging variants when doing the merge, because from the variants code from different tool may have a different breakpoint, but when doing a merge, you do union or intersections for only keep the region that overlap or the union.
Yeah, that's a good question. So actually, we do neither a union or an intersection. We will look at a window around all of the breakpoints that were discovered and try to find the best one using a statistical test.
Following the previous gentleman's question, that can GATK-SV able to detect functional translocations such as fusions that can be implicated clinically? And if it is not the best solution, what are other recommended algorithms or tools that we can apply to this DNA-based sequencing method? Thank you.
Yeah, so GATK-SV should be able to detect the translocation itself. SV annotate does not currently annotate gene fusions as one of its predicted functional consequences, but that's something we're really interested in trying to add in the future. I don't know when that would be, though.
Is there any weight system in GATK-SV? Weight system? Because the Explorer is so many.
Yeah, so we don't use a system to weight the different SV algorithms because we really, it's similar to discovering active regions and then reassembling them and genotyping using haplotype color. We're using the SV discovery algorithms to find candidate SV sites, and then we re-genotype all of them based on the RD, PE, and SR evidence.
Just specifically in terms of RNA fusions, gene fusions, if you have RNA, it's a very different story, much easier to see. And there's not a GATK tool, but there's a different tool developed out of Broad. The tool is Trinity, and there's a specific mode called Trinity Fusion to find gene fusions in RNA data. So a lot easier in RNA.
Okay. I think we are way over time, so okay, we're a little bit over time, so I'm gonna call it there for questions, and we'll come back in 10 minutes. What time is it? So we'll come back at 11:05.